been excited about your papers when they came out. So not only scientifically, it created a very nice vibe, but also how it came synergically. Instead of competition, you went for cooperation. So thanks for agreeing, and thanks for joining us today here. So I think Fred will go first. So Fred, before I leave you the scene, can you introduce yourself for a few sentences? And then when you pass to Caitlin, could you also introduce yourself with a few sentences?
Absolutely. So my name is Fred Heberle. I am an assistant professor in the chemistry department at the University of Tennessee. I got my PhD with Jerry Feigenson, and then I did a postdoc at Oak Ridge for many years, and then went to work in Ilya's lab in Houston for about a year, and then joined the faculty at University of Tennessee last year. So I guess I will go ahead and share my screen and get started.
Yeah.
Are you able to see the presentation?
Yep, it's perfect.
Okay, great. So thank you again, Erding, for inviting me to present this paper. And thanks to everyone who's listening in to hear this story about the use of Cryo-EM to directly image nanodomains in lipid membranes. So I want to start with the most important slide. These are the people that deserve all the credit for this work. My friends and collaborators and co-authors on the paper, Neil Waxham at University of Texas Health Science Center in Houston. And from the Leventhal lab, Ilya, Milka Doktorova, and Ali Skinkel, all of whom were at UTH, and me too, when we started this project a couple of years ago, and Hayden Scott, who was a postdoc working with me. Now, by the time this paper was actually published a couple months ago, everyone except for Neil had moved somewhere else. So Ilya is now at UVA, Milka as well. Ali is a graduate student at UNC, and again, I'm at the University of Tennessee, and Hayden actually just took a position at Oak Ridge National Lab. And I want to mention here that Ed and Alex are going to make a cameo in this story, so watch out for that. So my lab is interested in trying to understand the structure and the organization of lipids in the plasma membrane, like many of you are. And we're motivated by some of the unsolved mysteries of this very complicated organelle. So for example, we know that plasma membranes, and indeed many biological membranes, are composed of hundreds of distinct lipid species. So why is that? What purpose does that compositional complexity serve? Not only is the composition complex, but how these lipids are organized in the bilayer is also complex. Cells use energy to create interleaflet asymmetries in these lipids. And this is a beautiful example from Ilya's lab, from Joseph Lorentz paper that appeared recently in Nature Chemical Biology, where they determined the detailed asymmetry of lipids in a red blood cell. So there are asymmetries in the head group composition as well as the chain composition. And we don't fully understand why this is the case. And there are lots of groups out there, including my own, who are working to try to understand this. But the mystery that I'm going to be telling you about today is lateral organization. So when you have this many different kinds of lipids together in the same place, some of them aren't going to get along well with each other. And the intuition is that there's likely to be some non-random lateral organization of the lipids. And many, many observations point to that, both in model systems and in living cells or in cell-derived preps, like these giant plasma membrane vesicles. This is also work that was done by Ilya's lab, showing phase separation in GPMVs, but lots of great work on these systems from many other groups as well. So images like these raise lots of questions. What causes this phase separation? What properties of the lipid mixture control the size and the shape of these domains? Does phase separation happen in a living cell? Can cells control it? And how does this non-random organization contribute to function? So my lab uses a bottom-up approach to study some of these questions. We start with simple lipid mixtures. We try to understand their phase behavior, and then we add more complexity. One of the most useful experimental systems for systematically studying lipid phase behavior in a model system is giant unilamellar vesicles and fluorescence microscopy. So here's a typical experiment. You take 2 phospholipids and cholesterol, and you mix them up in all different proportions, and you add some fluorescent lipids to provide contrast, and then you just see what happens. So for many of these mixtures, you'll find coexisting phases over much of the composition space. At lower cholesterol concentrations, it's a solid fluid coexistence. But as we get up into physiologically relevant cholesterol concentrations, it's a fluid-fluid coexistence. So you have a disordered fluid phase that's coexisting with an ordered fluid phase. And these phases have very different properties, but for the rest of this talk, I'm just going to focus on their thicknesses. And as you're about to find out, this talk really is about thickness and how we measure it, and whether or not thickness can act as a contrast mechanism for detecting coexisting phases in cryo-EM images. So the focus is on this one property, thickness. Now, because the LO phase is rich in saturated lipids and cholesterol, it's thicker than the LD phase. And so I'll be saying a lot more about that. Now, an interesting observation made by many groups is that some 3-component mixtures don't appear to have fluid-fluid coexistence region. Here's an example. So you take this mixture, DSPC, DOPC cholesterol, but now you replace the DOPC with POPC. And what we and others have found is that there's no fluid-fluid region in this 3-component mixture when you look with optical microscopy. At low cholesterol concentrations, you still get the gel fluid coexistence, but once the cholesterol concentration gets above 10 or 12%, the GUVs appear uniform. So this is a very striking difference for a couple of reasons. First, DOPC and POPC are both low melting lipids, and they're not all that dissimilar structurally. So basically they differ by one unsaturation in one of the chains. But that one double bond is enough to abolish the macroscopic phase separation in the POPC mixture. The other remarkable thing is that POPC is abundant in the plasma membrane of mammalian cells, but DOPC is not. So this very striking difference in the phase behavior is something that's worth understanding. Now, an important clue came from looking at these same mixtures with FRET. FRET, of course, is sensitive to much smaller domains, in principle as small as a couple of nanometers. And with FRET, it was clear that the heterogeneity in the POPC system extended to higher cholesterol concentrations. So the same mixtures that appeared uniform with microscopy are clearly heterogeneous when we examine them with FRET. So probes that co-localize give a strong FRET signal that's consistent with non-uniform partitioning. Another way that we've looked at nanodomains in these mixtures is with small angle neutron scattering. And I don't have time to get into all the details, but the basic idea is that with neutrons, we can play games with contrast. So we can selectively deuterate some of the lipid components, and we can adjust the ratio of H2O and D2O in the solvent to match contrast. And so by doing that, we can tune the system such that when the lipids in the bilayer are randomly mixed, we don't see any scattering signal. This is scattering intensity versus Q and temperature for a uniform control sample. But when the lipids demix or phase separate, now we get a signal. And we can model that signal and pull out information about domain sizes and bilayer thickness. So this study used the same 4 lipids as the studies in the previous slide. But in this case, we gradually replaced the POPC with DOPC. In the previous studies, we found that DOPC results in macrodomains that are visible with microscopy, but POPC does not. And in the SAND study, we found that the ratio of these 2 lipids can be used to tune the size of these domains in large unilamellar vesicles. And we were able to correlate the change in the domain size with a change in the thickness differences between the LD and the LO phases, which we also measured with SANDs. So we found anywhere from a 6 angstrom to a 10 angstrom difference, depending on the ratio of POPC and DOPC. The greater the thickness difference, the larger the domains. So our conclusion is that the domain size is influenced by line tension at domain boundaries that in part arises from this thickness difference between the 2 phases. Okay, so everything I've told you up to this point is background information for the EM studies that I'm about to describe for you. A couple of years ago, I started talking to Ilya and Neil about using cryo-EM to image membrane domains. It's not a novel idea, but I think the typical thinking was, if this is going to work, it's going to require a probe. So something that's electron dense and that will partition between the phases or selectively bind to one of the phases to provide some spatial contrast. And you're going to see a beautiful example of that, I think, in Caitlin's talk. But considering what we know about the differences in the physical properties of LD and LO, the idea that this experiment could be done without probes didn't seem all that crazy. So maybe the thickness difference alone would provide enough contrast to distinguish these domains. And the reason is that with EM, you can actually see the layered structure of a membrane. And in fact, even by the late 1950s, the instrumentation and the methodology had improved to the point where membranes started to look like railroad tracks. So you can clearly see that in these images of frog neurons from a paper by Robertson from 1957. Now, as far as I can tell, these were the first really clear images of a plasma membrane, EM images. And so Robertson actually used these to propose a modified version of this trilaminar sandwich model of membrane structure. This was the structure that had been proposed by Danieli and Davson in the 1930s. So for Robertson's model, he interpreted the light inner region as the electron poor lipid bilayer, and the dark bands on either side as protein, 2 protein layers on either side of the membrane. We now know that that interpretation was wrong, and that the 3 layers here correspond to the hydrophobic core of the membrane and the 2 relatively electron dense polar head group regions on either side. Now, it's certainly tempting to think that the distance between those 2 dark lines corresponds to the head group distance in the bilayer. And if that's true, that means that we can directly measure bilayer thickness from a cryo-EM image. Now, I think it's worth mentioning here that membrane thickness is a fundamentally important membrane property in its own right. I don't think I need to tell anybody that's listening here that that's the case. So here are 3 examples of models that depend on membrane thickness. And I chose these 3, first of all, because they're well known within the membrane biophysics community. And second, because they illustrate an important point, which is that bilayer thickness itself is not a single thing. And that's obvious when you think about it. The membrane is a layered structure. You have head groups and then chains and then head groups on the other side. So you have to specify which layers you're referring to or what the reference points are for those thickness measurements. And you'll notice that in each of these equations, the appropriate thickness that governs the behavior is different. So in the solubility diffusion model, it's the hydrophobic thickness that governs the permeability. In the Lizotti equation that connects the area per lipid to the volume per lipid, it's the total bilayer thickness that matters. That's defined by the interface between lipid and water. And in the polymer brush model, it's the membrane mechanical thickness that relates the area compressibility and the bending rigidity through this relationship. So 3 examples, but there are many, many more. Bilayer thickness is an important parameter. So how is bilayer thickness typically measured? Well, the gold standard is a joint analysis of neutron and X-ray scattering data. That was pioneered by Wiener, White, and King in the early 90s. And then Kucherka, Nagel, and Katsaris and others later modified and extended that analysis to extruded vesicles. And this is one example where we used the Kucherka methodology to determine the bilayer structure for POPS. So this analysis relies on fitting scattering data to a model, but it's quite robust. And there are now structures for many different kinds of lipids that have different head groups, different chains, and at different temperatures within the fluid phase. The structural model itself is essentially a volume probability profile along the bilayer normal, where the different parts of the lipid molecule are described by functional forms like Gaussians. And among the parameters that come out of this are the full bilayer thickness. This is the Lizotti thickness from the previous slide, and the hydrocarbon thickness. So these are going to come up again later in the talk. Now here's a cryo-EM image of a DOPC vesicle that clearly shows this double line feature. So I'll tell you now, but I'll show you later that the distance between the dark lines in this image is about 29 angstrom. And that's remarkably close to the hydrocarbon thickness of DOPC at 25 degrees that has been determined with scattering analysis. So the question is, can we use this feature in cryo images to measure an absolute bilayer thickness? And the answer to that, I think, is maybe. So after working on this analysis for more than a year now, I think that doing this in a robust way is actually more difficult than it appears. And I'll show you why I think that's the case. But what I'll also show you is that these railroad tracks change in a very systematic way with increasing bilayer thickness. Thicker bilayers really do have a wider spacing, and regions of different thickness within the same vesicle are apparent even to the naked eye, at least in some cases. So I'll also try to convince you that the answer to this question, can we use cryo-EM to directly visualize nanoscopic domains, is yes. Okay, so a couple of details about the sample prep. All of our lipid films were hydrated in pure water for 2 reasons. First, salts and buffers can influence the quality of the ice during cryopreservation. And second, we previously found that it's much harder to make unilamellar vesicles in the presence of salt. And in fact, even when there's no salt, if you only have neutral lipids like PC lipids, it's also really hard to make unilamellar vesicles by extrusion. Here's some SACS data that demonstrates this for POPC that was hydrated in water and then extruded with a 100 nanometer pore size filter. So these Bragg reflections in what should be a unilamellar sample are a dead giveaway that there's actually lots of non-unilamellar stuff in there. But we also found that adding as little as 5 mol% of a charged lipid like POPG will increase the fraction of ULVs. So here's that data for POPC and POPC with 5 mol% PG. And here's a Cryo-EM image of DOPC vesicles that also have 5 mol% PG. And you can see that the vast majority of these vesicles are unilamellar. So we didn't use salt and we did use PG. So almost all of the data that I'm going to show you are from vesicles that have 5 mol% PG mixed in with whatever the lipid of interest is. And the other thing that I want to mention here is that all of the cryopreservation was done from room temperature. So the things that we observe in these samples, we hope anyway, corresponds to room temperature behavior. Here's one of those DOPC vesicles that's been blown up. I'm going to skip most of the details about how we analyze these images, but the basic idea is that we first find the contour of the vesicle, and then we take a small chunk of the bilayer, about 5 nanometers wide, and we bin all of the pixel intensities in the radial direction. And that gives us an average intensity profile in the direction normal to the bilayer. Now, if we average that profile over all of the segments in a vesicle and over many vesicles, it looks like this. So these are average profiles from about 60 vesicles. And there's a very characteristic W shape to this profile that you may have already noticed actually just from looking at this image. Our eyes are naturally drawn to the two dark bands, but those are actually flanked by lighter bands. And those are interference fringes, and they're a good reminder that this is really mostly a phase contrast technique. So we did this analysis for fluid phase bilayers of increasing chain length, and we found that the positions of the peaks and the troughs change systematically as these bilayers get thicker. And this figure shows the distance between the two troughs calculated for these bilayers of increasing thickness, of increasing chain length. And I've also plotted 2 of the bilayer thicknesses that were obtained from SACS analysis of the same sample. So in general, it looks like the trough distance in a Cryo-EM image, that's the black data points in the black dashed line. It looks like it corresponds to the hydrocarbon thickness. That's the orange line. And that was a bit of a surprise to us because we expected that as an electron scattering technique, the cryo would be most sensitive to the most electron dense part of the bilayer, and that's the head group region where the phosphates are. That's this value DHH. But EM isn't reporting the head group thickness, it seems to be reporting the hydrocarbon thickness. So the question that we wanted to answer at that point was, what is it that we're actually measuring here? And is this way of performing the analysis robust? So I hope you'll bear with me for a couple minutes here while I take you down a little rabbit hole. So at this point in the project, we got an idea that maybe we could calculate these EM intensity profiles from an MD simulation. And maybe that would give us some insight into where this profile comes from and how it depends on lipid composition or other parameters like the instrument settings. So it turns out that for many types of data, you can actually predict a signal from an MD simulation. We do it all the time with scattering data. All we need to do in order to predict the scattering signal is we just get the time average distribution of all of the atomic positions and that are projected onto the bilayer normal. And that's easy to calculate from a simulation trajectory. So once we have that, we multiply each of those atomic distributions by the corresponding scattering power of that atom, and then add them all up, take a Fourier transform, and that gives us a scattering signal. So if we use scattering parameters for X-rays, we get the corresponding scattering curve. SACS curve. And if we use the scattering factors for neutrons, we get the corresponding SANS curve. For neutrons, we can also pretend that some of the hydrogens are deuteriums, and that gives us a little bit different prediction for the scattering curve. And we can do that experimentally too, so that's useful. So the question now is, can we go from here to here? And if so, what's the relevant scattering factor, and what are the other operations that we need to do in order to get this W-shaped profile? So fortunately for us, somebody already figured that out about 15 years ago, and they wrote a nice paper that's pretty easy to follow. So I'm going to do my best to quickly summarize this. The primary mechanism for intensity contrast in the EM image is the phase shift of the electron as it passes through the sample. And the phase shift is proportional to the projected electrostatic potential of the object, which in our case is a lipid bilayer vesicle. Now, this potential is a complicated thing, but Wang and Sigworth decided to model it as a sum of two different pieces. The first piece they call the total atomic potential, and they estimated that as the sum of the Coulomb potentials of all the atoms in isolation. So obviously this doesn't account for how the electron density changes when the atoms are bonded together in the lipid molecule. So it's an approximation that we have to be aware of. So this first part is something that can be calculated directly from these simulated atomic number density distributions. That's the black dashed line. And then they added a second term, and that's the dipole potential of the bilayer. And that can also be calculated directly from the simulation. So when you add those two pieces together, that gives you the total phase shift profile in the direction across the bilayer. And that's the relevant physical property of the atoms that we needed to calculate these profiles. What about the other operations? Well, without getting bogged down in the details, the basic idea is that you first project that phase shift profile from a sphere onto a plane. And for a uniform vesicle, that operation is purely analytical. And then they have to smear this projection with a contrast transfer function. And that's analogous to the effect of a point spread function when you're talking about light microscopy. And what you'll notice is that the radial profile of this projected smeared image has that W-shaped intensity profile that we were looking for. But what you'll also notice is that the peaks in the original electrostatic potential profile that corresponded to the phosphates in the lipid head group region, those peaks no longer correspond to the troughs in the intensity profile. So not only are they not in the same position, but the distance between them isn't preserved during these operations. Now I'm going to get rid of all this math and make a couple points here. The first point is that for a uniform bilayer, all these operations are mathematical. They're purely mathematical. It's just a calculation that we do. The second point I already mentioned is that the contrast transfer function is analogous to a point spread function in optical microscopy. It basically just resamples the spatial frequencies in the image.